Variation in neurodevelopmental disorders.
Okay. Yep, that's on. All right. Thank you, Ryan. Thank you so much for the invitation to speak. I think this has been a great group. It's been wonderful to talk to everybody over the last 2 days. So what I thought I would do, after talking to Ryan a little bit, I understand you've had some technical talks, and I thought maybe everybody was a little seminar weary, and because of the group, I think everybody here understands genomics exceptionally well. So last night I pretty much took out all of the methods and tried to just give a series of vignettes on some of the sort of translational and functional genomic studies that we've been doing, and so we can talk methods, in the end if anybody's interested. But I thought I would talk about three things, starting with the work that we've been doing for a long time on structural variation in neurodevelopmental disorders, and just a short note on what we've been doing in prenatal diagnostics. Then really talk about some of the more recent studies that we've been doing in functional genomic studies of really highly penetrant mutations in autism. So, we've finally gotten to this point where we can start asking questions of these genes that we've been discovering in autism and neurodevelopmental disorders. How can we really understand what they do, and can we find sort of convergent final pathways in which they affect? And then finally, engineering genomic disorders, which is very new, and we are just generating the data. Some of it literally just came off the machine a couple weeks ago. So it's about half published stuff, and then the other half completely unpublished, and we'll see how it goes. Okay, so genetic variation, I'm going to refer to autism more often than, say, more global neurodevelopmental disorders or congenital anomalies, but really we study all of these. What we know, I think, is pretty clear that common variation is the major contributor to the genetic architecture of autism, although we've made very little progress in terms of finding actual common variants that are associated with the disorder. Rare single nucleotide variation from exome sequencing has been a sort of a boon of new discoveries in autism. Copy number variation has been studied for a very long time through chromosomal microarray, and in fact, at this point, chromosomal microarray is the recommended first-tier diagnostic screen for autism spectrum disorder. And then there's balance rearrangement. So translocations, inversions, balanced chromosomal abnormalities. This has been almost entirely ignored in autism, neurodevelopmental disorders, and really complex diseases more generally. The methods, and it's largely a technical barrier, and that the methods are still to this day relying on karyotyping, as they have been about for the last 50 years or so, other than new technologies for sequencing. So a lot of my work is really focused on this group of variants, and the reason is that when you have a balanced rearrangement that occurs de novo, you can disrupt an individual gene without concomitant loss or gain of genomic material, such as happens with CNV. So you can narrow in on a single gene of interest in a given complex disorder that you really don't have the opportunity to do when you have large gains or losses of DNA. Okay, so one of the questions we had, looks like the animation's off, is really, question is, can we change the risk? So what's behind this that you didn't see is, in terms of a de novo karyotype in a prenatal diagnostic setting, say, if you find a de novo translocation between the chromosomes on a karyotype, you carry about a 6.1% risk of an untoward outcome. And that risk estimate is based on cytogenetic studies done over 20 years ago. And to this day, that's still the risk estimate that we quote, 6.1% for translocation, 9.4% for de novo inversion. It has nothing to do with the actual bands of the chromosome that are disrupted, the genes that are disrupted, or any information on basically what the risk estimate is. So starting several years ago when I was a postdoc, we have been sequencing these chromosomal abnormalities and asking the question of, at sequence resolution, can we improve that risk estimate? And can we get to individual genes that might provide us more information? Most of what I'm going to talk about is using large insert jumping libraries, so I just want to describe them very quickly. Really, they had been around for quite some time, and I think made popular by this original study by Mike Snyder's group with Jan Corbel. We have been using them extensively. The idea here is, although genome sequencing has gotten cheaper, it's still quite expensive to cover a genome, and if you want to sequence structural variation, you really want a lot of coverage. So here what we do is we take the genome and we shear it up into very large fragments instead of small 200 base pair fragments. We then add an adapter to the end that we ligate to the end of each of those fragments, and we just designed a stuffer. So an internal biotinylated adapter that we can circularize the fragment on. We can now shear this circle, go in and grab that stuffer, and if we grab the junction site, what we've actually done by sequencing into the ends of that junction fragment is sequence a fragment that is separated by the size of the circle. In our case, usually we use 3, 4, 5 kb. So we now generate short reads on the end of a sequence that are actually separated by 3 or 4 or 5 kb in the genome. If we have a translocation or a rearrangement, we're then getting reads on each end of the fragment, and we've now narrowed a rearrangement down to the size of the insert. And depending on the coverage that we have, we can narrow it even further. The reason that I have really gravitated towards this method, you can't get point mutations. It's actually not very good for determining a lot of the genetic architecture of disease, but you can go very deep in coverage of the genome by using the spatial relationship between the reads rather than relying on the actual nucleotide coverage. Yeah, just okay. Oh, okay. So what have we learned from doing the sequencing? I'll give you just 2 small hits of past studies and then get into what we've been doing. So one of the things we found out from the original study where we sequenced 52 subjects with balanced chromosomal abnormalities from karyotyping was that about 20% of all of these events are actually complex with 3 or more breakpoints. And this was almost a 10-fold increase over what had been estimated at cytogenetic resolution. So it turns out that what we once thought were quite simple exchanges between the chromosomes are quite complex. And in fact, inversions are a pervasive feature of these. We found cryptic inversions in 90% of the complex events that we detected. So it led to this hypothesis that, in fact, inversions are mediating more complex rearrangements between the chromosomes. We also uncovered this extremely complex set of rearrangements that they had been finding in cancer at the time. This was in 2009 when we first found this first one, and the folks at Sanger had just published on this idea of chromothripsis, or chromosome shattering and recombination. And I was sitting on this data saying, well, I'm seeing this in these autism kids, except it wasn't quite the same thing. In the cancer genomes, you saw this oscillation of gain and loss of copy number variation within the shattered region. Here in these children with autism, what we're seeing is a significant number of localized breakpoints that are exchanged both between the chromosomes and within the chromosome. However, it's resolved to almost a perfectly balanced state. So in this case, you have about 23.5 megabases of the genome that's rearranged, and yet only about 5 kb of total genomic DNA gained or lost. So it's completely cryptic or undetectable by any method except for sequencing. Okay, so by using those methods, what we were able to do is essentially take these balanced chromosomal rearrangements that we knew, because we knew that they were going to be contributing to disease in a certain portion of the cases because they were de novo rearrangements in a child with a phenotype. We then sequenced them, got to the breakpoints, and in this one study, we found a series of 33 genes that were disrupted by breakpoints from balanced chromosomal abnormalities. Every one of these genes that are highlighted or bolded and italicized were actually new discoveries at the time that had not been associated with autism prior to that, such as CHD8 and SSTD2 and CAROL3. We then went and tried to interpret these single unique genomes, right? We have an N of 1 here. So in fact, quite a few of them could be red herrings. We did that through Convergent genomics. We looked at gene expression. We've built up quite a large CNV database of about 75,000 people at this point from diagnostic laboratories and controls. So we looked for a burden of CNVs. And in fact, we associated genome-wide association studies. So what we were seeing was an overrepresentation of common variants from genome-wide association studies in autism and schizophrenia over the rare variants, the genes that we were detecting by rare variations and structural variants. It was also quite interesting that we saw this overlap between psychiatric disorders and children with neurodevelopmental abnormalities that disrupted the gene compared to psychiatric disorders where you saw this burden of common variation at the genes. And I think one of the main things that we found were these individual gene contributors to genomic disorders. So these were large segments of gains and losses of DNA that were previously referred to as genomic disorders. And what we found is with a balanced rearrangement, you can disrupt this one gene in that region and recapitulate much of that phenotype. Okay, so we've gone on and done this many times, and this is some unpublished data. We've sequenced 174 karyotypic abnormalities at this point. We've actually sequenced more at this point. It turns out that still 21.4% of them are complex rearrangements. So clearly we're missing this with karyotyping in the current standard of care. And we actually find that the level of convergent genomics is actually quite strong. So it continues to turn out that when you have a de novo balanced large rearrangement of the genome, the genes that are disrupted tend to be quite strongly enriched for causal factors and neurodevelopmental abnormalities. One point I want to make, and this is where I'll get back to this in a second, but in every one of the BCAs we've sequenced to date, I think, at least it was this way at 150, I haven't gone back and looked again, at least one breakpoint differed from the karyotypic interpretation. So of 174 cases with a karyotypic rearrangement. Not once have we sequenced them and actually found both breakpoints to localize to exactly what was predicted by the karyotype. It's always off by at least a subband, and in many cases a band or more. So I'll come back to that in a minute. Okay, so what have we been doing since? All right, what happens if you don't have a karyotype? What is the impact of structural variation in autism? So this was a study that we started a few years ago, right when the copy number variation work was being done in the Simon Simplex collection, which is a collection of idiopathic autism patients, sporadic autism patients from trios. So we have parents, an affected sibling, and there's actually a quad for a lot of them, an unaffected sibling. And both exome sequencing and copy number variation was being done by chromosomal microarray. We proposed to sequence probands. We wanted to sequence trios, but we couldn't get the funding to do it, so we proposed that we sequence probands with jumping libraries to ask the question of what is the structural variation landscape beyond just copy number variation? So what do we see beyond just copy number variation? So we wanted to really characterize the full spectrum of structural variation in the autism genome. Here's just a look at the overall metrics. So, this was the method I was describing. This is the look at the coverage. So on average, we generated about 100x coverage of all of our libraries. So, I mean, this is extensive coverage compared to, say, the 1,000 genomes, right? I'm completely amazed that Ryan and these folks were able, with 4x nucleotide coverage, to get the results that they do. So we generate 100x insert coverage. You can see this is sort of the insert size distribution of the libraries. They're right around 3.7 kb, fairly tightly distributed between 3 and 4.5 kb. And so that's an important note. So we have a threshold, right? So we have an insert size of 3.5 kb. So say less than 5 kb, we really can't tell the difference between a normal read and a deletion. Okay, so I'm sorry, I did forget to take this method slide out, but fine. So if we look at our jumping libraries, we have developed a number of methods. We use read depth algorithms and clustering algorithms and a combination of multiple clustering algorithms. But it was really using the inserts with read depth that I think has made the biggest improvement over just plain clustering, like everybody else has been looking at these things. And so if we look at the arrays that have been run in the Simon Simplex collection, we actually do quite well at recovering what they find in anything above, say, 100 kb or 200 kb, 240 kb. We find all the deletions, and even at 40 kb, we pretty much hit most of the deletions and a high proportion of the insertions. And our pipelines are pretty much tailored towards diagnostic work. So we worry a lot about false negatives. And you can see that, potentially we're overcalling by quite a bit. So this is the proportion of the total variants that we call that are actually seen on the arrays, as opposed to the portion of variants on arrays that we can pick up that are gold standard array calls. However, what we've gone on and now tried to validate, we've been validating a lot lately. And it turns out that, somewhere around 85% of the things that we call as CNVs that the array missed actually turn out to be valid. So, we're still missing a chunk of this, but we are picking up a good bit. And I think, as we've all known, the arrays turn out to be quite fallible in a number of places. And of course, then we're just below the resolution of the arrays a number of places as well. Okay, so what have we been finding? I think this is one of the more interesting things that we found from the Simplex collection. And it is that there are duplication signatures in the genome. So as we were sequencing along and having the experience of chromotrypsis already, we were used to kind of putting together these complex rearrangements. What we found is that a relatively high proportion of duplications in the genome that were detected by array turn out to be fairly complex rearrangements. And one of the more interesting classes that we found was this signature of 2 duplications flanking an inversion. And in fact, we turned out that we found a number of duplication signatures that you can see in the genome. And in fact, you can pick up on arrays, and we found them in diagnostic practice as well, and I'll show you those in a minute, where essentially you have 2 duplications flanking a balanced inversion. And we would call this dupe INV dupe. It's similar but completely distinct from the dupe trip dupe variants that Jim Lupsky has described for microarrays and done a lot of work on. Effectively, what this is 2 duplications in proximity to each other that flank an inversion. And we found this signature in 9.6% of the autism probands we looked at. And then if we consider other complicated duplication signatures, we actually see them in 12.5% of the autism subjects. So a significant proportion of autism subjects actually have this signature complex variation. And as I said, about 5.6% of all duplications detected by chromosomal microarray turned out to be complex rearrangements. So this is the signature that we find that turns out to be quite common and had not been described before from population-based sequencing studies. But effectively, what you get is 2 duplications in proximity an inversion in between them. So you end up with then a head-to-tail duplication signature, a tail-to-tail duplication signature, and a large inversion. And you can see this is a microarray. You clearly got the 2 duplications, but what you obviously can't see is the inversion in the middle. We have gone back to a large number of microarrays and looked at them, and this signature actually pops up quite a bit in different samples, where you get 2 duplications in proximity to each other. Of course, they're just interpreted as benign duplications, and they can be small, they can be large, but what's missed is the fact that they're actually underlying a quite large inversion. So we published the first one of these in a study we did on early onset neuropsychiatric disorders, where we saw this dupe inversion dupe signature that actually disrupted a gene. What was interesting about it is when we went to clinical diagnostic array data, what we found is this duplication signature was present in this number of cases. So I mean, it's rare. It's something like 0.1% of cases, but you can clearly see this duplication signature on diagnostic arrays. So basically, anybody who has an array study, go look at your arrays, and I'm quite sure you will see this kind of a pattern in a certain number of your cases. Okay, what else did we find? Well, we found these dupe signatures that actually also underlie clearly pathogenic events. This is a de novo large pericentric inversion that disrupts OTS2, but the chromosomal microarray pattern actually was just 2 benign small duplications on the chromosome, and yet we know OTS2 is a pathogenic locus in autism. We saw other signatures that look like paired dupe signatures, but in fact, they involve 3 duplications with inversion in the middle, duplication with deletion and inversion, and even a duplication with a nested deletion. So if a sequence is duplicated and then you have a nested deletion, you in fact go from copy number 3, copy number 2, copy number 3, and the middle portion actually does not involve any gain or loss of material. Okay, so in summary of these really complex duplications, where we're characterizing the breakpoints, and we're pretty much done with them, but a large portion of them are microhomology mediated. You might have guessed this. It suggests some sort of microhomology mediated template switching. I had a good time talking to Tom Glover today and thinking about this. We see insertions at the breakpoint a huge number of times, again, suggesting some sort of template switch that's happening. We find gene fusions, predicted gene fusions, only 9% of the time, and in fact, hemizygosity of a gene, only 9% of the time. More often than not, the gene is simply duplicated, and that makes the interpretation of this disruption in the disease quite hard. Because you can imagine you can have the gene inverted and duplicated, but if the entire gene is duplicated, you actually haven't disrupted the message, you've just gotten another copy of it, unlike the OTS2, which was completely disrupted. Okay, so in autism, and this is, please don't hold me to this, this is sort of our working numbers, and they're yet to be explained, but I think this is really the take home, is we know the common variation is going to be a major component of the disorder, and Bernie Devlin has done some nice work with a large consortium to show that maybe it's around 49, 50%. While the de novo exome variation has certainly gotten a lot of attention and been remarkably helpful for us to characterize the disorder, best estimates are maybe it accounts for 5% of cases that have a de novo loss of function mutation. In the Simons collection, and this number of 6.7%, so we think that somewhere, it might be somewhere around 15%. We're not done with the validation. We don't have all of the inheritance status for all these, so please don't hold me to this, but at this point, our estimates are around 15% of the etiology is due to structural variation. In the Simons collection, and again, these are sporadic, they tend to be less severely affected than some other populations, particularly populations that go into clinical diagnostic laboratories that might have a higher burden of intellectual disability. The arrays actually picked up about 6.7%, so we think this is almost doubling the yield of the arrays by simply looking at small variants that are below array resolution or these kind of complex rearrangements. So we think this was sort of a useful strategy. We want to now do much larger scale trio analyses, but effectively what we see is small deletions. We actually found reciprocal translocations. We found those complex rearrangements I told you about. We've actually found chromothripsis in the sample, but absent any karyotypic abnormality, so very small variants moving around. We see insertions into genes, and then small cryptic CMBs, all that were missed by previous characterizations by microRNA and exome. So if you kind of extend this thought into the clinic, you can make the assumption that as we push exome sequencing and we push microarray, we're still missing a certain proportion of the genetic variance that is quite detectable with our technology right now. Okay, so that transitions into genetic diagnostics, and I'll keep an eye on time. What time are we officially done? Okay, I can just kind of hammer through a couple of things, but okay. Genetic diagnostics. So I don't want to belabor this too much because this is old, and we were talking about at dinner actually last night that we had sequenced all these genomes, and yet this you know this paper was one, and yet it became a publication simply because of who it was. So this was a case that had multiple congenital anomalies and a translocation that came into the clinic, and based on the methods we had developed, they asked if we could do on a research basis. So they signed in a research study if we could provide a prenatal diagnosis of the case based on having a karyotype. So we developed our pipeline we basically just squished the pipeline down into a 2-week turnaround where we were able to make jumping libraries, do the sequencing, throw our computational pipelines at it, and Sanger sequence validate the breakpoint within 2 weeks. And what we found was the translocation actually disrupted a known causal locus, CHD7, which is the cause of charge syndrome. So this child obviously went from a 6.1% risk of an untoward outcome based on no knowledge of the breakpoints to, a very, very highly penetrant locus that was actually disrupted. We made a predictive diagnosis of charge syndrome at birth. The clinical diagnosis was charge syndrome in this case, and unfortunately the child passed away at 10 days of age. It was a very severe form of charge, which is, I'm sorry, everybody probably doesn't know, charge is a multi-system developmental abnormality that involves cardiac defects and intellectual disability and a number of other defects, including an undescended testes, which they were able to pick up at 32 weeks on the ultrasound, but you wouldn't be able to pick up at 12 weeks when they originally did the ultrasound and found a cardiac defect. So we have gone on and done this. That was the original case. We have gone on and done this a number of times in the clinic and in concert with the clinicians in the clinic. I'm going to tell you about just one of the series of, I think, 10 we've done so far. And it's because it's sort of an interesting case, and it feeds into some of the developmental work we've been doing. I mean, we haven't published any of this, but this was a case where a child got a karyotype of an inversion of chromosome 8. And it happened to be a quite savvy mother, and she's asked us to tell the story, so we tell it, that had seen me give a talk at a meeting. And she was given a risk of 6.1% based on the de novo inversion. And she has clearly said that she was risk averse and was likely to terminate the pregnancy, but had seen that she knew that we would be able to sequence. She understood genetics fairly well, so she wanted this done. So on a research basis, the sequencing was done. We found an inversion that disrupted this gene, KHD-RBS3. It's an RNA binding protein. It's involved in alternative splicing. Seems like something you should probably have 2 copies of in the genome, and yet through our Convergent genomics, we saw deletions of this gene in controls. We saw a series of disruptions of the gene in exome cases. And so our interpretation was absolutely a variant of unknown significance. Certainly, we weren't increasing that risk beyond what it would have been otherwise. And so she changed her mind and decided to go forward with the pregnancy based on these results, and you can make your own interpretation on that decision. But about a year ago now, Julian was born, and he's perfectly happy and healthy, and he's home, and the mom has given a number of testimonials and gone through these processes on how this sort of influenced her decision in the counseling realm. From a research standpoint, the question is, well, I guess first of all, from a clinical standpoint, the question that we used to ask is, can we do this? And clearly, yes. And in fact, just a month or 2 ago, we did one in a week. So we had a 7-day turnaround from a karyotype to a breakpoint sequence. And then the question, of course, comes up, and this has been really widely debated in diagnostic practice in general, and prenatal diagnostics more specifically is, should we do this? And I think as a conservative reflex test, at least to conventional cytogenetics, it offers superior resolution to make a clinical medical decision. And I think the key point that I'm sort of stuck on is that medical decisions are being made every day right now with incorrect information. So I just told you that every karyotype that's ever been given to a mother is, most likely off by a subband, or at least the ones that we've sequenced so far. So at the very least, at least we're giving them the information of where these breakpoints actually occur, and then the risk that comes with that, of course, everybody interprets differently. So the point of telling you this, though, is to tell you about the experimental stuff that we've been doing in cell-free-fetal DNA sequencing. I don't know if you guys have heard of Jacob Kitzman, but he wrote one of the really fantastic papers on this. Yeah, I know. It's weird. No, I knew Jacob, and he actually helped us. But there was all this great work going on in non-invasive prenatal sequencing. Jacob and Jay and those folks had narrowed down the point mutations and looked at the exomes and done haplotype resolved work in a trio. What we wanted to do nobody even still, although there was one translocation detected by a company, but in general, nobody had looked at small CNVs and certainly not a balanced rearrangement in cell-free fetal DNA. So the idea of cell-free fetal DNA, is everybody familiar with this? That you have DNA circulating in the bloodstream of the mother from the fetus, and so if you can take that DNA and circulating in the bloodstream, there's small shards of DNA. You can sequence it, and you can basically do a diagnosis if you have a copy number abnormality, but specifically an aneuploidy, without doing an invasive test. So what it's really useful for, though, is trisomy 21. And beyond trisomy 21, you know, its sensitivity and specificity and positive predictive value is all over the place. And if it's not an aneuploidy, it's very hard, and it's not actually all that useful. Well, I just showed you that a huge portion of autism and other abnormalities have nothing to do with trisomies or aneuploidies. They have to do with much smaller variants. So we asked the question of, could we find this inversion? So basically, Jacob and Jason Jury made a library for us a few years ago now, and we sequenced it extremely deep. We generated 1.7 billion reads. We got 52.6x haploid coverage. Now, effectively, the circulating cell-free fetal DNA is a mosaic of the mother and the child. So what this ends up being is 38x coverage when we sequence the small shards, and it's 24% fetal DNA in there. So about a quarter of the reads come from the fetus, 75% of the reads come from mom. So we then went and asked ourselves, can we find this chromosomal rearrangement just from the circulating DNA in the fetus? And the answer is yes, we can. And the only reason that this hasn't become a widespread test in genomics is because we can't really. So we threw all of our methods at it. The jumping libraries jump right over the breakpoint quite nicely. So, you know, we get 68 reads on one chromosome or on one end and 37 reads on the other. But it turns out it's sitting right on a line element. And so no matter how deeply you sequence this cell-free fetal DNA and try to align these small up to 140 base pair fragments, we never got a clean split that we could pick out sitting on the line on one side. Now, if we go in and just write a little program to grep every read in the FASTQ file because we know it's sitting there, we pull them out. So they're in there, but there was no way we could actually find them. So I mean, out of an abundance of caution, we didn't publish this, and we still haven't published it because while it's a positive finding, there's no way we could have actually done this blindly, and nobody has done it blindly since. So I think this is a real challenge. So what we are doing, though, is we've now really generated a large number of jumping libraries on the mother. We have the father, extremely deep coverage, and we have the proband, deep, and jumping libraries. And so what we're trying to do now is characterize what structural variation can we find from cell-free fetal DNA. You know, at least what proportion of the structural variant spectrum would we be able to find from circulating DNA in the mother that could obviate the need for invasive, high-risk clinical testing during pregnancies. Okay, so that's the end of the structural variant stuff. What I wanted to do was talk a little bit about functional genomics now and some of the work that we've been doing that I'm pretty excited about in terms of characterizing these variants. So I'll tell you the story about one gene, CHD8. This was one of those 33 genes that we found. It's actually interesting. It was a gene that was in a microdeletion syndrome. And a couple of years before we found it by our translocation, it was, you know, there were a number of genes in this microdeletion syndrome. It's a fairly rare microdeletion syndrome. CH8 was one of them. So we hit it by a translocation somewhere around 2009. And it was just one of this pile of genes that, you know, meant nothing. There was almost nothing published on it in terms of disease association. And it was this N of 1, which is the problem with sequencing a new genome is you don't know much about the gene disrupted. Since then, of course, there's been an explosion of papers in autism, all of which have effectively replicated CHD8 as one of the most penetrant. De novo loss of function mutations of CHD8 is one of the most penetrant causes of autism. Much more recently, there was a study of tumor types, 21 different tumor types from exome sequencing from the Broad and others, and they actually found CHD8 as one of the genes disrupted in a pan-cancer analysis from mosaic loss of function mutation. So, and there have been a couple of other papers suggesting perhaps a role for chromodomain helicases in general and CHD8 specifically in cancer. Okay, so the point of this study was, all right, so we had this gene that we knew was fairly penetrant. And the exome studies that had been published, particularly one, you know, was really pushing these pathways and networks that the genes that harbored de novo loss of function mutations were part of. And these are from kind of curated PPIs, and the networks are fine, and there's been this assumption that it leads to disruption of the beta-catenin network, and then this pathway that leads to autism from there. The only problem is, I mean, this is all predictive, right? We don't actually know what happens when you knock down CHD8. So what we wanted to do was really try to perturb the network of genes that are regulated by CHD8 and see what actually comes out at the end. So we designed a study where we had the CHD8, we had iPS-derived neural progenitor cells, so early neural precursors. And what we wanted to do was knock down CHD8 expression in early neural precursors from a control with an isogenic background, generate an allelic series. We did this a large number of times because we did transcriptome profiling, and of course, it can be noisy. You really need to model the variants from MRC, because as you guys all know more than anybody. We also did a zebrafish study, because one of the main phenotypes that have come out of the papers to date on CHD8 disruption is macrocephaly. Okay, so this was the study that we did. This is a look at the series of shRNAs we used. So we got a little obsessive over this. We actually used 6 independent shRNAs to knock down the gene 6 different ways in 6 different lines. We did it all in duplicate. We took one of the shRNAs and ran it again, so that we actually had this one in triplicate. We then took shRNAs targeting genes that aren't in the genome and ran those in duplicate as comparisons. So we had the native line, we had the shRNA treated line, and then we had the control lines. This is the level of knockdown that we observed, so it clearly worked, and it ranged from 40% to 70%, exactly what we would expect in a hemizygous individual with a loss of function mutation. And then when we actually look at the protein levels, we see that they've gone down, just as we would have predicted. Okay, so what did we find? Well, you know, one of the debates about CHD8, it's a chromodomain helicase. It remodels chromatin, and it was thought to have repressive functions, and yet there was also this other literature on activating functions. And you know, that's very much what we see. So if we look at the genome and the results I'm going to show you, this is at a nominal p of 0.05. If we go down to a q of 0.01, 0.001, actually the patterns hold the same. So I'm just showing you a larger swath of genes. But basically, the functions of CHD8 seem to be more repressive than activating. So you see more genes go up when you knock down CHD8 than you see go down. Fine. The pathways that you look at across all the differentially expressed genes have pretty much nothing to do with neurodevelopment. So here's a gene that we know contributes to autism if you have a loss of function, yet we have pathways that aren't all that related to neurodevelopment. But it turns out if you look at the upregulated and downregulated genes, all the downregulated genes are related to cell adhesion, synapse formation, axon guidance, neuron differentiation, all of the pathways that we had been searching for in autism and neurodevelopmental disorders. The genes that were upregulated were more to do with kind of transcriptional regulation and cell cycle maintenance. We did see an association of intellectual disability genes in that group. But in general, we asked the question then, and you know, of these genes that go up and genes that go down, what does the autism profile look like among those genes that are regulated? Is it the case that actually CHD8 regulates a large number of autism genes, and that's why we see this association? And in fact, if we take the genes that have been curated, there's 628 genes that have been curated by the Simons Foundation for Autism Research as having been implicated one way or another in autism. It's a very broad set of genes. It's not terribly specific, but we just asked a very broad question: are those genes associated with CHD8 gene expression? It turns out if you look at the upregulated genes, they're not at all. If you look at a reduced set of genes that have stronger evidence, they're not at all. But if you look at a broad set of TCGA cancer genes, and this is 6,000 genes that have been implicated in cancer in any way, shape, or form, again, not very specific, you actually see a quite significant enrichment among upregulated genes. Yet if you look at those downregulated genes, you see a really strong enrichment of autism genes and not cancer. So there actually seems to be this specific enrichment of genes depending on the repressive or activating function of CHD8. We then looked at ChIP-seq. So we took antibodies and did ChIP-seq, chromatin immunoprecipitation sequencing. We used 3 independent antibodies and only looked at the peaks in which each antibody was bound to a region of the genome, and we got a Q less than 0.05 on our ChIP-seq peak analysis. So this is really a very tight set of genes that are replicated 3 times with 3 independent antibodies. And what we find overall is an enrichment for CHD8 binding sites in the upstream region of the gene near the transcriptome start site. In fact, if we look at the epigenetic roadmap data, there's a very, very strong enrichment for active transcriptional start sites for CHD8 peaks. So repressive function, activating function, where it binds, all of this certainly suggests that a large number of genes in the genome have some CHD8 plays some role in activating or repressing them. You know, that role based on whether it's the regulatory role that it plays might be tissue specific, but CHD8 clearly binds to a large number of genes in the genome. In all three antibodies, we actually see 7,300 peaks. If we look at any one antibody, we see about 1,200 peaks. And this result was replicated by Jim Noonan's group in different cell types. So I'm telling you this because I want to give you some insight into what happens when we integrate data sets, when we integrate functional genomics data sets. So I just told you about the genes that went up and down. Well, what happens when we put the ChIP-seq data? So what if you're bound by CHD8? So these genes up here are indirectly downregulated by CHD8 because they're not bound at all by CHD8. These genes are indirectly upregulated. These genes are directly regulated in these two corners. And then these genes are bound by CHD8 but not regulated at all. And then here's the list of all those CHD8 boundaries. Well, it turns out that if we look at cancer, be it the TCGA cancer set or this more restricted set of genes that had de novo somatic mutations from Lawrence et al. More recently, or a cosmic cancer set that includes point mutations and CNVs, they're all statistically significant in genes bound by CHD8, but not genes regulated by CHD8 indirectly. And in fact, if you just look at the binding sites, the TCGA results are extremely significant, as are the point mutations. None of the autism sets are significant in the ASD bound genes if you consider those safari genes I told you about. But there's one thing that's special about the safari genes, and that they weren't just discovered by genetic variation, they were also discovered by neurobiological hypotheses and functional studies. So genes involved in synaptic formation and that were studied based strictly on neurobiological hypotheses and model systems are also included in that set. And in fact, when you look at the indirectly downregulated genes, so not bound by CHD8, that's where that signal was coming from. On the other hand, if you look at genes that were disrupted from de novo loss of function mutation studies in autism, they're actually enriched among the CHD8 binding sites. So these are two different sets of genes that are both implicated in autism by CHD8. And it depends on how you look at CHD8. In the pathways that came out of the initial exome sequencing studies were chromatin modification, transcriptional regulation, and synaptic regulation. And it looks like CHD8 touches each of those pathways, but in very different ways in its regulation. And the reason that this is important is if we then go look at brain span data. So this is co-expression module. This is gene expression data across developmental timing, from prenatal to postnatal. And work done in papers published in Cell last year showed that, in fact, there are co-expression modules of genes that are strongly enriched at various developmental time points. And one of the strongest enriched time points was early neuronal development, when you had the de novo loss of function mutations that were enriched for chromatin modifiers. And in fact, what we find is the genes that are bound by CHD8 are strongly enriched in exactly those same time points, whereas the other set of genes that are indirectly downregulated by CHD8 and associated with neuronal differentiation and synaptic formation are actually associated with the co-expression modules that were found later in development. And if we layer CHD8 data over a second study that did the same thing in brain span, we find the exact same pattern. So what we find is this sort of master regulator of many, many different genes actually touches on a number of different pathways that have been associated with autism neurodevelopment. And we weren't able to sort of tease those apart until we actually looked at the integration of multiple different functional genomic data types. Just the zebrafish, I'll tell you quickly, had macrocephaly. How's that? And Evan Eichler had done a study where they actually looked at patients with macrocephaly and found the exact same thing. Macrocephaly was one of the strongest associations from our ChIP-seq data. So this suggests both model organisms and iPS-derived cellular models point us towards pathways that are very important in autism from one strong effect. And that's exactly what we show here, is the developmental timing, whether CHD8 is actively regulating a gene or indirectly regulating a gene, may be differentially influenced, and the pathways that are implicated may be differentially influenced in autism. Okay, so the last 15 minutes, I will show you something totally new that we've been doing. And this is using genome engineering methods to derive genomic disorders. And I think this is something that's it's been fun in the lab. So we had done some studies similar to the CHD8 work where we did RNA sequencing of 16p11 microdeletion syndrome. So genomic disorders are among the most common causes of autism and neurodevelopmental abnormalities. They are recurrent CNVs, copy number variants, both deletion and duplication, where they are gains and losses mediated by the allelic architecture of the region, which is segmental duplications flanking the gain or loss genic region. So 16p11 is one of the most common ones. It's been associated with a number of different disorders, and you have 29 genes here in this genic region and 10 genes here in these segmental duplications, in which you have 2 copies of them. So of course the mechanism is a mispairing of these segmental duplications. So you either end up with a loss of the genic segment and 1 segmental duplication, or a duplication of the region and a duplication of 1 segmental duplication. So the problem with NHR mediated rearrangements is, although they occur they're the most common class of copy number variants, they have recurrent breakpoints. So you can't actually get to the individual driver genes within the region. And yet, despite the recurrent breakpoints of 16p11, this is one of the most heterogeneous phenotypes that we have with strong effect genes in neurodevelopment. In fact, it's been associated with neurodevelopmental abnormalities, autism, schizophrenia, other neuropsychiatric disorders, as well as BMI, obesity, and various anthropometric traits. We did some work in zebrafish in this paper published in Nature a couple years ago with Nico Katsanis, where they knocked down every gene in the region in zebrafish and found a strong effect driver of neuroanatomical phenotypes. But we wanted to ask the question, genome-wide, what does the transcriptome look like associated with a CNV? And so we did RNA-seq on a series of multiplex families with deletion or duplication of the 16p11 region. We also looked at mouse cortex of a model that has deletion or duplication of the syntenic 7qf3 region that was created by Aaliyah Mills at Cold Spring Harbor. And I'll just basically give you the overview of this because it's just an intro into the CRISPR work. But effectively what we find is if you have a deletion or duplication in an NAHR-mediated region, we find no dosage compensation whatsoever. So in 16p11, if you're deleted, all the genes go down. If you're duplicated, all the genes go up. Maybe that's not surprising to most of you. We actually were hoping to find some dosage compensation and perhaps rule out some of the genes in the region, and we couldn't do that at all. And in fact, the segmental duplications behave fairly well as well. When we looked genome-wide at the transcriptome, we found a number of signals, but basically what we found is the genes within a CNV region are the most significant genes by far in the genome. So the genes that are actually directly implicated by the CNV in both mouse and human are by far the strongest signals in the genome. And it's a little unfortunate because one of the things we would have liked to have seen was a number of loci that had actually strong regulatory effects. And we don't see that. And I think, this is a little bit underpowered and we want to do this on a much larger scale. But the one interpretation that you might take from this is the effect size really has a ceiling genome-wide on kind of the regulation that a given CNV can have. One interesting thing we found was positional effects. And I think this is interesting because of it might have an impact on sort of nuclear architecture and physical interactions between the chromosomes. And in fact, what you see is, okay, so if you take the genome and bin it up into all possible 1 meg bins, and then ask, do we have an enrichment of differentially expressed genes in any of those bins compared to elsewhere in the genome, the region itself implicated by the CNV is by far the strongest in both mouse and human. However, among the most significant bins on the end of the distribution is the 16p distal region. So just distal to the 16p11 microdeletion syndrome. If you go look in mouse, you see the exact same region is actually statistically significantly enriched for differentially expressed genes. But the proximal region isn't at all. It's right smack dab in the middle of the distribution of the entire genome. So you have one side of the CNV that's enriched for differentially expressed genes, and the other side of the CNV that isn't at all. Our colleagues at Lucerne University, Alexander Raymond and his lab have now explored this, and they have microarray data, and we replicated each other's data sets on this. And effectively, what happens is if you do 4cc and you use the focal point of 16p, the 16p11.2 region, the 600 kb region, or this 220 kb region that has a distal 16p11.2 CNV syndrome that's been associated, but it's much more rare. It turns out that there's quite a few physical interactions between these two regions. And of course, they're in close proximity to each other anyways, but it turns out that each of the genes, and I'm showing 2 here, but there's actually quite a few others, that have a physical interaction are actually differentially expressed in our analysis. If you then go genome-wide and look at some of those bins that were on the significant end of the distribution of significant bins genome-wide that seem to be regulated in trans, and you overlay high-C data on it genome-wide, there were a number of peaks where you actually found a bin that was differentially expressed, and you also found a high C peak, suggesting that they're in close proximity to chromosome 16p11. So these data suggest, and there's a lot more work to be done here at much higher resolution, that perhaps the gain or loss of the region actually leads to a shift in the positioning of the chromosomes and could lead to physical positional effects both in cis and in trans. Okay, finally, the convergence on common pathways. So I showed you the CHD8 data. A number of other genes that have been associated with autism have converged on those same pathways. And then when we look at the mouse cortex, what we see is cell adhesion, regulation of transcription, and neuronal differentiation. And those are the exact same genes that have come out of the autism exome sequencing studies, and they're the exact same networks and pathways that I just told you about in our CHD8 study. So I think at least early on, in a field where we had very little to hold on to for quite a long time in terms of, bona fide causal factors, in autism, we're starting to make some serious progress in terms of genes, pathways, and networks and how they're regulated and how they may influence developmental timing. So what we then wanted to do was ask the question, okay, we have these CNVs. Can we model them? It's not ideal to have patients with different genetic backgrounds one at a time, and we have to get peripheral tissue, and we have to look at lymphoblastoid cell lines. And it turns out that the lymphoblastoid cell lines only express about 60% of the genes that were interested in autism, whereas the neural progenitor cells and the mouse cortex had 80 to 92% of the genes. So we wanted to know, can we model microdeletion syndromes in a dish? Can we create them in a dish? So we had done a series of genome editing studies with CRISPR-Cas9. I'm assuming everybody knows about CRISPR and Cas9. The idea is we design a guide RNA targeted to the genome. We use a Cas9 endonuclease to create a double-strand break. You rely on non-homologous repair. To repair that break in a certain percentage of the time, that will go wrong and leave you with a small indel or a point mutation. So this has been used extensively. I don't know if I took out the structural variant slide. Okay, yeah. So it's been used generally for single guide RNAs to induce point mutations. So in fact, the idea is you have a guide RNA, it targets a site, you then have Cas9, you end up, it leads to non-homologous repair, you end up with a small indel or a single nucleotide transition or transversion. We've also done some studies. There have been other studies in the field, and then we just completed one where we show that you can actually create larger rearrangements using a dual guide method in which you target two different regions. They simultaneously cut the DNA, lead to non-homologous repair between the sites, and you get a deletion. We have actually done, we did a study where we sequenced the whole genome. We did another study where we did targeted analyses. And it turns out that this is a fairly efficient process. So up to, depending on the guide RNA that we chose, somewhere between 25 and 50% of the time, we could actually make a deletion. 3% of the time, what we actually made was an inversion. So just by using a dual guide, and I took out the slides for this, but just by using a dual guide cut site, 3% of the time, you actually get an inversion of the region between the two guides. And in fact, we induce translocations a small number of times as well. And we figured this out by sequencing. We did targeted sequencing to 3,300x coverage. So every nucleotide in the on-target and off-target site was sequenced almost over 3,000 times. So we can kind of count every molecule that was disrupted. And it turned out to be quite interesting. You also get the point mutations that you expect with single guides. So when you treat something with a dual guide, kind of the idea is anything that can happen does happen. When the genome is forced to repair itself, you get all kinds of funky rearrangements. So knowing that, we wanted to ask the question, can we actually use this for 16p11? And genomic disorders, more generally, it's just that we understood 16p11 and had all the data for it anyway. And so this is data that has just kind of come off the sequencer within the last couple of weeks. But effectively, we wanted to model this in iPS cells and then differentiate those to different types of cells. We tried the dual guide strategy where, okay, you have these segmental duplications, right? So remember, we have these two segmental duplications. And if you put CRISPR to the unique regions within them, you can probably induce a deletion between those two segmental duplications. So we designed one guide RNA to this region. We designed one guide RNA to this region. They're unique guide RNAs. They only blast one place in the genome. The idea being you can then come in and cut them and lead to a deletion of the unique sequence. But that's not actually what happens in vivo, right? It's a mispairing, and you actually lead to a gain or loss of one of the segmental duplications. So an ideal model would be to take out the genic region as well as the segmental duplication. And that would be a recapitulation of the in vivo model. So we did identify, we used the degenerate blast against the segmental duplications, came up with one region that would target each segmental duplication, but nowhere else in the genome. And the idea would be that we would cut the segmental duplications. Instead of non-homologous repair, we would induce homology-mediated repair, just as occurs in vivo. So we had this sort of debate among us. I had no doubt that this dual guide approach would work. We had already seen it work a bunch of times. The idea is we would generate basically a 575 kb deletion of the internal region, leave the seg dupes intact. It's not ideal, but it's pretty good, and I thought we could do it. We weren't sure if we could do this method, where we would actually target the seg dupes and get them to recombine on their own. But if we could, then we would be left with a 740 kb deletion, as well as one copy of the seg dupes would be taken out, and we would have one copy of those genes. One of the investigators on our collaborative team actually bet me that we would make duplications as well. And this is actually what occurs in the cell, right? You get a mispairing and a deletion or duplication. I didn't think that was going to happen, but there's only one way to find out. So we did the experiments. Basically, we designed the CRISPR. We targeted the seg dupes. We actually did FACS sorting after we generated the CRISPR treatment so that we could get the single cells, because we didn't want a heterogeneous population of cells. We're in the middle of doing genome-wide copy number analysis, but we have looked at the region itself with a qPCR, and then we've now done genome-wide RNA-seq. Okay, so here's what we got. The 575 kb deletion that we tried worked, and the 740 kb deletion worked. Here is the control line. This is a copy number. This is just a qPCR analysis. This is the native line. This is a patient with a 16p11 deletion. This is a patient with a 16p11 duplication. This is the 575 kb and the 740 kb deletions. And in fact, we did end up making duplication. And this occurred pretty frequently. So 13% of the time we were able to get NAHR-mediated deletion. 4.3% of the time we were actually able to generate NAHR-mediated duplication. When we then went and looked at this genome-wide, we had all this RNA-seq data in vivo. So we looked at our models. We generated an RNA-seq study in which we did the CRISPR-induced deletions and duplications, the native control lines treated with Cas9 and not a guide RNA to see what the Cas9 treatment might do. And then we looked at patients. So we had deletions and duplications in patient lines. We did RNA-seq across the genome. This is a look at the chromosome 16 region. And what you see is it turned out quite nicely. So all the deletions cluster together. They cluster with the patients, and the families cluster together, but the 740 kb deletions and the 575 kb deletions also cluster together. All the controls cluster together and with the normal mother, and all the duplications cluster together. When we look at the region itself, it looks exactly like I had shown you that the in vivo state looks like. Basically, all the deletions go down. This is RNA-seq data. All the duplications go up. And within the segmental duplications, the duplications go up, the deletions go down, and these 575 kb deletions disrupt basically all the genes within the region and none of the genes within the segmental duplication. So it looks like we have actually induced NAHR-mediated repair that disrupted one set of genes in the segmental duplications and the genic region itself. And this is a look at the genes within the segmental duplications. And there's actually, when you normalize them to the patient, it looks like the patients are a little higher, but you absolutely get deletion and you get duplication in the CRISPR models that match the patient lines quite nicely. So if we then think about that genome-wide picture that we got from the human 16p11, we've done the same analyses in the CRISPR, and we get the exact same results. Again, underpowered, we don't have that many samples, but effectively what we see is the 16p11 deletion is, again, the most significant region of the genome. So this clearly has worked. It's worked in the number of cells that we've looked at, and we can then model it for statistical analyses. And we're now moving on, but we think that this is basically a model that can work for all microdeletion and microduplication syndrome. So we think we can now model NAHR-mediated rearrangements in addition IPS lines and then differentiate those into all the lines that we might be interested in moving forward. Okay, so conclusion. Cryptic and complex structural variations are a component of the etiology of ASD. They're not an explain all, but they certainly do have a role in autism and neurodevelopmental disorders. I think whole genome sequencing can really provide a high resolution reflex test. Conventional diagnostic methods, there's a lot of debate in how pervasive sequencing should be in diagnostics, and that's a big debate. I'm saying on a very small debate. I think this offers a high resolution option for particularly for pregnancies in which we can give them the exact breakpoint, and then those decisions can be made after that. And finally, NAHR-mediated genomic disorders can really be modeled by careful targeting of segmental duplications And I think there's the opportunity to model structural variation more broadly using new genome engineering technology. So thanks for your time. I think we finished right on time. And these are the folks in my lab that did all of this work. These are just a few collaborators. There's many, many more that I wasn't able to list. And these are some of the people that have funded this research. Happy to take any questions. Please. Mm-hmm. Autism spectrum. Yeah. 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 Right. No, it's a great question. My guess, the guess, is that in the Simon simplex collection is actually a low proportion of cases compared to, say, if you had a sample loaded with intellectual disability, congenital anomalies, or dysmorphism. And I'm guessing that because, you know, where we see 15 to 22% CNV burden in general cases that come in for neurodevelopmental, due to neurodevelopmental disorder for microarray testing in the Simons collection, where very careful microarray was done in the entire family and the sibling, you only see a burden of about 6.7%. So I think this is, and this has been widely debated in the Simon simplex collection. I'm not telling you anything that other people haven't discussed, which is that, you know, we think that there may be some ascertainment for less severe cases in this collection. And I think the genetics are bearing that out to some extent. So my guess would be that it actually is a little higher than what we're predicting with the structural variation load and more severe neurodevelopmental disorders. But we're going to in schizophrenia and other neuropsychiatric disorders, I'd bet it'd be lower. Yep. Exactly. Right. Yeah. No, in schizophrenia, I'm guessing it's going to be lower, and we do know that the CNV, if we follow CNVs, and I think that's what the structural variation stuff will do. The complex stuff I'm finding is probably a good parallel for the CNVs. You know, the burden's lower in schizophrenia and bipolar disorder and ADHD, but it's there. It's significant, non-trivial. It's a component of the etiology, but not as strong as in early on. I think that's going to follow, but again, that's a prediction. Next couple of years, I think we're really going to figure that out. Yeah, good question.
Yes. Just curious, in your studies, have you come across with this order, autism, there's any difference from region to region, let's say China to the United States?
So I have not dug terribly deeply into the epidemiological data. I mean, I know it, and in general, it's fairly stable, but does bounce around. I don't know specifically China, if that's your question. I know schizophrenia is extremely stable across populations. The incidence of comorbid affective disorders and mood disorders bounces around quite significantly. And you know, there's always been debate whether that was really a diagnostic thing. So, for example, in the U.S., it was the last numbers I looked at, somewhere around 30% of cases with schizophrenia had a comorbid mood disorder, and yet in India it was like 5%, in fact 3%, and so that seems to be a diagnostic thing based on the studies that have been done. Autism, I can't say for sure between China and the U.S. I know that the prevalence has been rising. You know, everybody knows this in the U.S. and other developed countries. I probably can't speak to the underdeveloped countries as well, but it's certainly the prevalence is certainly going up, and you know, quite significantly. While the prevalence of intellectual disability and
Did you find any effect of stress?
we haven't looked.
You have.
No, we haven't. Our research is very much genetic.
Thank you.
Sure, but there are people doing that.
Yeah.
I can tell you in various groups, they're absolutely very interested in correlating genetics with some of the phenotypic features that are out there. My lab.
Yeah, I'm sorry. I just, to follow up that question, there is the famous cohort of the famine in China where there is an increase in schizophrenia. on women who were pregnant, and so that most of the fetuses had spontaneous abortions because the starvation was so profound. But the ones who didn't, that might be a very interesting cohort for you to really understand perhaps the stage and the environmental causes possible.
Right. So
for schizophrenia, I have no idea about autism.
exactly. The thing about autism, you know, the heritability is fairly high. Of the component that we've shown, these are de novo, what we are finding so far with the tools that we have, right, which is almost certainly picking off the lowest hanging fruit and not a major component of the disorder. And I think that's what all the research shows so far. You know, these are de novo loss of function mutations of these really sort of important genes, chromatin modifiers and transcriptional regulators, and these genes that control the regulation of all these other genes in the genome. I suspect, and again, this is a little bit of speculation, but based on what I just showed you with the developmental timing and how they're, you know, extremely early in developmental timing is when they're most highly expressed, I suspect those are just really severe cases that the environment isn't necessarily going to be terribly influential. But as I showed you in that donut, it doesn't explain that much of the variance. So I do think there are these other components that could be quite important, and you know, we haven't been studying them, but at some point that will be the next step to do. We're still kind of looking for the low-hanging fruit at the moment. Anybody else? Yep. Mm-hmm. Right. No, it would. I think. Well, I think that's part. No, it's fine. It's because I went through like 100,000 slides in 3 seconds. So that was really the point of these studies. And they were published in Cell a couple of years ago, or I'm sorry, just last year. But basically, this data is basically, and that's it's just the beginning of your question. You know, so I think you're asking a very big question. This is a small component of it. But basically, you know, what they were finding is you can track gene expression signatures along developmental timing from kind of very early to postnatal and in different in different brain regions, right? The gene expression signatures, and we shouldn't overstate this because there's a lot of genes that are expressed the entire way, right? But the gene expression, the co-expression of genes did differ. And when they differed, the genes that had been associated with autism by de novo loss of function mutations were more likely to be in various co-expression modules. And the chromatin modifiers and transcriptional regulators were more likely to be early. And so that's kind of what this is getting at, is those, you know, highly regulatory genes were more likely to occur early in development. And when we looked at our CHD8 genes, the ones that CHD8 bound were also more likely to be in these co-expression modules. So it's certainly, it's a, you know, and this is the effect size I'm showing you, which I guess I didn't explain before. So it's a relatively strong effect. But it doesn't, you know, it doesn't explain everything, but it is certainly a statistically significant enrichment. Whereas the genes that were involved in cell adhesion, synaptic plasticity were a little later in development. And you know, if you want to just spitball and throw out hypotheses, perhaps those are the ones that we should be targeting for kind of, you know, therapeutic ends, because it might be that these early ones, things have gone pretty bad pretty early. But later on, you know, it's more subtle phenotypes. And so I think people are trying to get at that. And, you know, that's one of the purposes for going back and finding these patients that have these mutations and figuring that out. Get exactly the question. You know, if you want to start to think about treatment of pharmacogenomics, things like that, you know, maybe these are the genes that we should be targeting. But that's how. Yep, absolutely. Oh, of course. Yeah.
So what's going on with the large number of bound but non-regulated? I mean, it looked like the number was much greater than.
Right. So, and this is replicated. So this is, if nothing else I told you is true, this one is. That is basically even in kind of 3 antibodies in our study, and then and from Jim Noonan, he looked at mouse brain and embryonic stem cells. We are looking at RNA-seq in one tissue, though, right? So it could, you know, this is a chromatin modifier, a chromatin gene involved in chromatin remodeling. It may absolutely be that it's regulatory in other tissues, and if we did RNA-seq in a bunch of different tissues, we would find different genes turning on and off that it's bound to. But it's absolutely the case that a small portion of the genes that it's bound to in this study, again, there might be power issues and all that, you know, are differentially expressed when you knock it down. Perhaps that was an expected outcome looking back on it, although we didn't think it going in. because there's no way CHDA is actually that strongly regulating, half of the transcriptome, right? Even if it's bound there, a third of the transcriptome. So that said, they are enriched for autism genes and cancer genes, right? Pretty significantly. And, they are also, I showed the bound to transcription start sites, a vast majority. So I suspect they're important, and CHDA is important for those genes, and we just don't understand why. I feel like if it's a painting, we're just looking at such a small piece of it, and it's frustrating. But clearly, there's enough statistical enrichment that's unbiased to suggest that they're important. Good question. Okay.
Let's thanks, Mike. Thank you very much.
Thanks. Thank you. Thank you so much for